Today's lecture will be discussing chemical reactions and enzymes. So you can see below I bulleted all the main points of the lecture, so in your notes, as you're taking notes, you can pay special attention to these points. So everything that happens in an organism, whether it's growing, interacting with its environment, reproducing, or moving, all those things are based on chemical reactions. So if you can see, that's me down there reeling in some big giant fish this summer, and I had to use my muscles to do this. Muscles contracting are a chemical reaction. So this lecture is going to explain a little bit of how those chemical reactions inside our body work. So a chemical reaction is a process that changes one set of chemicals into another by changing the chemical bonds. So the things that go into a chemical reaction are called reactants, and the things that are produced are called products. So for example, as carbon dioxide enters our blood from our cells, it reacts with water to produce carbonic acid. So if you look at the picture below, we see carbon dioxide coming from our tissue into our blood, and it's reacting with water. So these would be the reactants of this chemical reaction because they're the things going into it, reactants, and they're producing carbonic acid. So they're combining to form carbonic acid. So chemical reactions need energy to take place. So the activation energy is the energy that is needed to get the reactions started. So if you look at the graphs below, you can see down here it says reactants, and up here it says products. So in order for the reaction to happen, to get from reactants to products, we need some sort of energy here. So this point here, they call this the threshold. This is the amount of energy that's needed for this reaction to happen so we get our products. Now, sometimes reactions, chemical reactions, can take a long time to happen. And our body is constantly moving. We can't sit around and wait for these reactions to take place. So we have enzymes. Enzymes catalyze or speed up chemical reactions that take place in the cells. So they help these reactions go faster. So if you look below, this little purple guy here represents an enzyme. And the reactants of an enzyme reaction are called substrates. So these are the molecules that go into the reaction, the thing that the enzyme works on. So that would be this little yellow guy here. And they bind to the specific site on the enzyme called the active site. So this would be right here. So that's where it binds to, and it's going to have a specific shape for every substrate. So each enzyme only works on one particular substrate, so its shape is going to exactly fit that one particular substrate. Kind of like a puzzle. Nothing else is going to fit. And then what's produced in this reaction, again, is called products. So we have the substrate binding to the active site on the enzyme. The enzyme speeds up the reaction, and in this case it's breaking this molecule into two, and we have two products that are produced. So these enzymes help us function properly by helping speed up all of these reactions so we can function the way that we need to. Enzymes speed up chemical reactions by lowering the activation energy. So remember, that was the energy needed for a reaction to take place. So if you see in blue here, without the enzyme, this reaction needed this much energy. But with the enzyme, it only needs this much energy. So that reaction can happen way faster with the enzyme than it could have without the enzyme. So again, it's just making these reactions go faster. So here we have an example of how this works. So this is called the enzyme substrate complex. So the reactants of the enzyme catalyzed reaction are called substrates, so we already know that. So in this example, it's water and carbon dioxide. So those are the things going into the reaction. So this little green guy here is the enzyme. So these substrates, the carbon dioxide and the water, bind to the active site on this enzyme, which is specific for only carbon and oxygen. Oh, only carbon dioxide and water can bind here. And the reaction takes place, speeds it up. The substrates are converted into the products, which in this case they're put together to make carbonic acid. So that is the product of the reaction. Here's just one more example. This is a sucrase enzyme. Enzymes are always named with an ACE at the end, A-S-E, so you can always tell when you're looking at a molecule if it's an enzyme, it ends in ACE. Just a helpful fact. So this one is acting on the substrate sucrose. If you remember from the carbohydrate lab, sucrose is a starch, a carbohydrate. So this one is digesting that starch. So the starch goes into the enzyme, binds to the active site. The enzyme is breaking it down, converting it into its products, and you can see here a glucose and a fructose molecule are produced. So again, those are monosaccharides, if you remember. So what this is doing, this is how we digest a starch molecule, a sucrose molecule. So like I said before, the substrate binds to the site on the enzyme called the active site. So the active site has a specific shape that only one substrate can fit into it. So each enzyme only can act on one particular substrate. So it fits kind of like a lock and key. So the substrate would be the key. Only one key fits into this lock, fits into the active site for the enzyme. So it's specific in that kind of way. And just a little helpful information, enzymes are usually named for the reaction that they help, which is a helpful way to identify them. So a protease is an enzyme that breaks down proteins, lipase breaks down lipids, polymerase breaks down, or DNA polymerase breaks down DNA. So again, it's always going to end in ase, and it usually starts with the name of the thing that it's getting broken down. So proteins, protease. You can kind of figure out just by looking at it what that enzyme is going to do. And lastly, enzyme activity is regulated by a couple different factors. So temperature, pH, and different regulatory molecules. These are all factors that affect the activity of enzymes. So for example, the human body has a temperature of 38 degrees Celsius. So most of the enzymes in our body work best at those temperatures. If you tried to do some sort of enzyme reaction that normally happens in a human body and you're doing it in a very cold place, maybe you're doing it in test tubes in a very cold lab, it's probably not going to work as well because it needs to be at the proper temperature. Same thing works for pH. So several digestive enzymes in the stomach, our stomach is very acidic. So a lot of those enzymes only work in very acidic conditions. So these sort of things, temperature, pH, can affect how the enzyme works. So this is all for this lecture. If you're confused about anything, please go back and rewind. Don't forget to submit your summary from your notes onto the Google form that's attached to this assignment on Schoology. Okay? Thank you very much, and I will see you on Monday.